Environmental DNA is pretty much everywhere. How could something that, you typically might see in a crime scene be useful on a farm? Typically in the past, if you wanted to go out and, maybe you wanted to see what insects are in that bush over there, what fish are in the river, it's going to take quite a large ecological survey to go out there and, put your nets out, put some traps out. You're going to need someone to sit there and identify every single little bug that you've got, and it's incredibly time-consuming to do.
Where it helps us do effectively the same thing is where we find the worm or we find the snail, we find also that organism's DNA. So the same inference that we would make on its occurrence by seeing it, we can still make the same inference by just picking it up with the same DNA. It's the same essential fundamental idea.
Once you start thinking about the space in which you live and the kinds of things you want to collect, then that's how we want to start tailoring the sampling mechanisms to collect something that's relevant.
So you've got to then know a bit about the biology of the animal or the plant that you're looking for as well to think, all right, well, whereabouts is this DNA likely to be?
This is the first of the eDNA sampling sites. We're actually on the Moore River. This is part of the Yued Aboriginal Corporation. So the Regan's Ford is this particular location. And interesting, the results that came back from this. You've seen the data?
Yeah, so from my understanding, you took air DNA and water DNA here. So,
yeah, correct.
So if you've taken a water sample here, it's going to show a composition of a whole lot of DNA that could have washed down from up in the catchment. So what we detect here, it doesn't necessarily mean the species is at the site, but it could be somewhere upstream that's flowing down. We found a few neat little species. So we found the oblong turtle. There's the western pygmy perch and the night fish and freshwater cobble. And you also did air DNA where you hung up an air sample in the trees over there.
Yeah.
And this would detect all sorts of things like pollen. I mean, even us standing here with our skin coming off us can be picked up into the wind and be immersed in the air. It's an amazing amount of DNA that is just blowing around all around us all the time. So there was Eucalyptus rudis, which is the flooded gum.
Orange flooded gum, yeah.
Yeah, so that one there was in the air DNA, and when you're sampling the air, it could be from wind as well. I don't know how windy it might have been on the day and how far that it could have possibly blown, or it could be somewhere that's quite local in here. So that's where you got to link up what you detect in the DNA with your local knowledge and those living on the property. I guess probably an uncertain classification was the brown or grey teal. One of those teals doesn't live in the area. It's actually endemic to New Zealand and is not going to be here in WA.
Not likely to be here in WA.
But this is one of the things with DNA is that we can sequence what we detect, but then you've got to try and match it up with what we know about different genomes of different species. The barcodes don't match exactly, so it might pick a more closely related cousin that's within the area.
Might need some further investigation.
Yeah, exactly, some further investigation.
Adam, this is upstream from where we were at Regan's Ford. So we took both water and terrestrial roller samples here at this point. Unfortunately, since the sampling was taken, you can see that we've had a bushfire through this site.
The water sampling, again, same with what we've got downstream, is that it's representative of the broader catchment.
Yep.
It's everything that's aggregated, washed off land or what's in the river, and we collect in the sample. And then we can see what the difference is between what we collect here versus what we collect downstream.
Yep.
If a new species arises between point A and point B, this species here has occurred.
Yeah.
So then you can narrow down your range that you're looking at.
Yeah.
The other method that we that was used here was the roller DNA.
Yeah.
So basically like a little paint roller, and you can use that for terrestrial DNA. So just standing here, again, our DNA falls on the ground when the kangaroo hops somewhere, and or a lizard goes from A to B. It leaves in its path a trail of DNA.
Yeah, right.
Which you can detect with the roller. It's got to keep in mind that you could also be picking up stuff that could have blown in from the air, but primarily it's looking at movement on the ground.
Gotcha. So good at picking up, mammals, reptiles. How long will DNA stick around for?
It depends where it is.
Yeah.
So, sometimes it can degrade, a day or two, sometimes it can be a week. When it's drier, it will last a lot longer than when it's wetter.
Okay.
It really depends on the conditions, and that would be supporting bacteria to break down those cells and that DNA.
We had a sample response to a eucalypt species, very much on the threatened / endangered species list. In fact, there's only, I think, 3 other sites in Western Australia where it's currently been identified.
So that was the Midlands gum?
Yep.
Yeah, when you look at records, there's only 3 known locations that are actually recorded.
Yeah.
Whereas this is a reasonable distance from where those records are. Alternatively, it could also represent a misclassification with threatened species. They tend to get more focus and funding to get their DNA sequenced.
Yep.
So, but if there's a closely related cousin that hasn't been sequenced, that could also, you could be picking up that. The only way to find that out is go for a walk through the bush and see what you can find out.
Yeah, looks like we've got another field trip on
our
hands
I think
coming
so.
up
Yeah,
for that
right.
one. Yeah. This is a very different landscape to where we came from the Yued Aboriginal Corporation sites at Regent's Ford and Magumba.
Yeah, very different.
So we're in a different catchment, different soil type, very different landscape. Like this is a reasonably intensive beef cattle production system.
Yeah.
But interestingly, a very similar hit as far as the samples on those species. So
the mix of species.
The mix of species. I spoke to the owner here, Dave Humphries, and I said, we've got a sample or a hit on the turtle, the oblong turtle, and he goes, oh yeah, he said, I've seen the predated shells. He said, I've seen live animals. I know the owner, in the discussions I've had with him, is really keen to see what we get out of this data and then what he could potentially do at a farm management or a habitat conservation planning.
I mean, and certainly even just looking around, I mean, you can see parts of where, you're talking about a clay bottom, soft bottom creek, but with some collapsing banks. So then that's where we need to then look and say, okay, well, whereabouts are the turtles likely to be laying their eggs? Could they benefit from more bank stabilisation?
Yep. This is a site one of the sites that we took a water sample on here at Dwyer's property. We've identified that the pygmy perch was picked up here. Yep. But unfortunately not the oblong turtle. It doesn't seem like he's come upstream from Humphrey's place next door where we found him on the creek. But I don't know. What are your thoughts about this site?
That is an interesting finding, and from what we heard before, that the turtle have been up here before, but about 10 years ago.
Yeah.
So my first thought is thinking, okay, well, what happened upstream here around that 10-year ago mark? And it would be worth looking at satellite imagery, aerial imagery, to see, okay, well, what happened to the habitats in and around here, particularly the smaller tributaries coming off here that are similar to the one that's downstream.
Yep.
So maybe there's been some sort of change. Another thing that comes to mind is that it could also reflect a shrinking population. So the turtles are not going to travel any further than they need from the downstream source location.
Yep.
When they're coming up, then they need to. So if the small population could be sustained by the tributaries downstream, then it's a waste of energy to come up any further.
Yep, makes sense.
My experience is that I don't have time often to be delving into all the little nooks and crannies of nature on my farm. I'm very busy trying to keep the animals alive and trying to keep, the fences up. So to be given information that then allows you just to get on with that issue rather than have to sort of do all the research, much better.
Now, with it being a lot more available, there's sort of 2 broad approaches that you can do with eDNA. One is where you can be looking for a particular species of interest, but also for pests and invasive species as well. It's useful for that detection to say, it's early detection. Can we detect their DNA before we even see them? Because by the time you see them, often that's when the populations are so large, it's getting hard to control, that's right. But if you can get them early, the other approach is the broader meta-barcoding, which basically means that they look at all the DNA that's in the system or within a particular group of animals to help you say, well, what's here on the farm? And then you can say, all right, out of those that we have detected, which ones are most threatened? They really need it, and say, oh, well, what is it about that species? And within this area that's likely to be constraining that population, and that's your low-hanging fruit, so you get better bang for buck.
I was attracted to the idea of, new technology and what, you could learn from it. And, in discussion today, we're talking about using the DNA to test the soil for the microbes. That's a fascinating subject. That's exactly what I'm trying to do, is change the growing environment to improve the nutrition of the animals and, improve the life of this farm and improve the quality of the farm. And the microbes are the essence of it all.
One of the biggest and strongest predictors in my model of ecosystem health, which we know already for wetlands, is the extent to which there is buffering vegetation around them. It doesn't have to be 250 metres of vegetation, it just needs to be a little bit. That little bit of vegetation, if it's got any foliage, creates shade, which then reduces evaporation, provides habitat for a whole bunch of different things. And that level of habitat complexity is a really simple way of increasing the worth of these actually potentially very useful biodiversity oases in agricultural landscapes.
There's obviously a lot of growing awareness around improving conservation, but there's also a lot more market pressures these days too. Particularly
from a natural capital perspective, that's really becoming quite a commercially viable sort of avenue now for a farmer.
Particularly out of Europe, when they're buying food, they've got very conscious consumers and they want to know that their food is coming from somewhere where the landholders are really looking after the land, and the food's not at the expense of the biodiversity and the natural landscape. And eDNA is one of those ways that you can sort of then say, well, we are doing our part. These are the threatened species that we do detect on our property. And here's the different things that we're doing to help support those and keep them there.
Talking about natural capital and being involved when you don't have immediate results, I think that's where eDNA was interesting, in that you do get immediate results that potentially can improve the way you're farming, and then that contributes to improving your natural capital, because you're creating a better environment to grow commercially, but you're also creating a better environment for everything.